Hello, everyone. Welcome to the welcome to the first ever SciFilmIt competition live show. Over the next hour or so, we'll be watching 8 wonderful short science films, and we will award prizes as decided by our expert science film jury. Now, just a note for anyone who might be watching on the SciFilmIt website, if you are on there and you want to join the conversation or you want to ask questions, then you can jump over to the YouTube channel or the YouTube live stream because there'll be a chat in there so you can put in your questions for the filmmakers and maybe for us as well. So I'm Robbie. I'm one of your 3 hosts this evening, and we'll begin by introducing each other. So first up in Barcelona, I will introduce Eleonora Aquilini. So Eleonora has a curious and creative mind. She's a scientist turned communication specialist. She has a PhD in microbiology and environmental biotechnology. She spent more than a decade in several labs around the world investigating that which is invisible to the eyes. She published her last scientific discovery in Nature Microbiology this year. She obtained a Master in Science Communication and Future Media at Salford University. And since 2020, she's been Communications Director of a global biotech company based in Barcelona. And she's also a proud human to a Black Labrador, and she loves music and dreams of traveling around the world. Thank you so much for being here, Eleonora.
Thank you, Robbie. Thank you for the lovely introduction. And it's my pleasure to introduce Enrico from Boston at the moment. Enrico is an eclectic scientist and science communicator whose mission is to advance human knowledge and to create safe spaces where science and storytelling meet. This has resulted in his PhD at the PFL in Lausanne, Switzerland, and in several science films. Now he is a postdoc at MIT, and his research focuses on the study of roughness evolution during fictional processes with numerical simulation. And it has been translated, his research in the science film Wearing Well, presented at the New York Imagine Science Film Festival in 2018.
Thank you, Eleonora. And now back to Robbie Price, who's in Geneva now. He's a bee scientist, filmmaker, and educator. He works with academic institutions and research projects around Switzerland to support the creation of impactful storytelling through film. He's passionate about bees, reimagining education, and exploring how story can support the quest for accessible information. Robbie is also a member of the Swiss Young Academy.
Thank you, Enrico. So we're going to be the 3 hosts this evening. We'll also see some of the jury, and we'll also see some of the winners. So before we get into the films, I would like to introduce SciFilmIt a little bit. So SciFilmIt is an association, and it was founded in 2019. It is an evolution of the Exposure Science Film Hackathon project that was launched in 2016 in Lausanne. And our goal is to use filmmaking or the filmmaking process to unite the worlds of science and story. We devise and support science film projects in Switzerland and more recently across the world. And we've run our hackathon course in Swiss universities since 2016, and we currently have 6 universities in Switzerland involved, and also 1 outside, which is very exciting new development. The hackathon, which of course is different from what the project that we're talking about tonight, but I'll touch on it slightly. It places artists and scientists in teams over a 3-day period, and it takes them through a very intense process. They come up with a scientific idea, a narrative, they write a script, plan production, and shoot and edit a film in just 3 days. So the evident goal of this 3-day project is to make a film. But actually, the most important thing for us is the process that these participants go through. The challenges that they have in, first of all, getting to know each other, working together in a team that has very different skills, different knowledge, different bases of knowledge, and then kind of working together, coming up with all the solutions to all the problems that come along in the process. So that's the true value of the hackathon, and it's something that we're really proud of. So to speak now about the competition that you're all here to see this evening. Last year was obviously a year like no other, and we were less able to create the hackathon events. And so we launched our first science film competition to get scientists and artists collaborating from their homes. We received submissions from 3 continents, on a huge variety of subjects, and 8 films were selected by a committee, and then these films were sent off to the jury for them to decide which ones were the best. So to move on to our jury of expert science film experts, we have, just going to go to my notes very quickly. So the jury is made up of 4 experts based in Switzerland, India, and the UK. And our first expert was Morgane Freund, and she is a Swiss filmmaker with a deep interest in the creative spaces between art and science. She now studies at HSLU in Lucerne and creates documentaries about nature and animals. Her last project, Memoirs de Méduse, is an experimental film about jellyfish. Thank you so much, Morgane, for being part of this project. Prasenjit Yadav is a molecular ecologist turned National Geographic photographer and explorer. Prasenjit is one of very few photographers who integrates science deeply into his photo stories. He chooses ignored subjects, landscapes and species, and finds ways to develop engaging and accessible photos. So those are our first 2 jury members. We also have Professor Kirsten Prushoff, who is on the next slide. And Kirsten is a co-founder of SciFilmIt, and she's a multidisciplinary scientist at the University of Geneva. She's an educator with a passion for communicating science across disciplines and audiences. We also have the wonderful Theo Blossom. Theo is a passionate conservationist, creative science communicator, and an award-winning filmmaker and photographer. Theo is fascinated by how people connect to nature. He's equally interested in how science is communicated and made accessible to different audiences. So those are the 4 jury members who watched all the films and assessed them. And we send a big thank you to all of them for their time, energy, and when we spoke to them after they'd seen the films, they really were very impressed by all of the films that they watched. We, of course, couldn't do this without sponsors. So we have to say a big thank you to the University of Geneva, with whom we have worked for about 3 years now, and we hope to continue working with them, and Nikon, with whom we've worked for 4 years, and these are incredibly important in all our projects. So thank you very much to both sponsors. I will now pass over to Eleonora.
Yes, thank you, Robbie. It is my pleasure actually to introduce the screening for the finalists of the Swiss category. We will see 3 beautiful short films, On the River, Solar Fuels, and finally the mysterious organelle. So I remember you that if you should have any question for the filmmakers, please write them in the YouTube chat, and we will have the conversation with the winners in which we can actually ask your questions. So now relax and enjoy, and good luck to the participants.
Sei still und hör dem Wasser zu. Dem Nebel, dem Tropfen, der Welle. Der Fluss ist alt und weiß mehr, als wir wissen. Es gilt zu hören, was er sagt und wie er sich bewegt in seinem Lauf. Danach richten wir uns, wenn wir klug sind, am Bach wie am Strom. Wir sehen es an und staunen immer neu. Die Hydrologin, sie hat es mit dem Logos, mit dem Wort, spricht also mit dem Wasser. Und der Geograph schreibt achtsam auf, was das Wasser in die Erde schreibt, was es bewegt und ändert und verlagert, wo etwas aufblüht und gedeiht und leben kann und auch was untergeht in diesem Wasser, aus dem wir gekommen sind. Du lebst davon und du kannst darin untergehen. Es trägt dich und es trägt dich in die Welt und unter alle Sonnen und zieht dich dorthin, wo für dich kein Atmen ist. Seine Schönheit und sein Zauber, sie sind unbegreiflich und zugleich erbarmungslos. So mächtig sind die Widersprüche und seine Erscheinungen zwischen Bewässerung der Felder, einer todbringenden Dürre oder einer Überschwemmung und dem Erstarren in Eis. Wie immer du es bestaunst oder besingst oder in einer herrschsüchtigen Hybris gar zu regeln versuchst, dem Fluss bist du ein Nichts. Und inzwischen wissen wir auch, wer die Musik eines Wassers reglementiert, wird selbst ertauben und sein Mund wird trocken werden. Warum ist es, wenn du am Ufer hockst, stets so jemand wie ein Therapeut für dich? Therapon heißt ja Gefährte. Sei einfach still und höre zu. Der Welle, dem Tropfen, dem Nebel.
Welcome to Solar Energy, yeah, with Connor. On today's episode, we will be discussing solar fuels. What? Yes, solar fuels. Specifically, we are going to be talking about hydrogen. So, let's get started. Enough energy hits the Earth every hour from the sun to power the world for 1 year. I hear you saying, Connor, stop, you old fool. You expect us to believe something so outrageous? Oh, my friends, I do indeed. You can even do the calculation at home or check resources like this one. But how can we harness this energy and make it useful for us humans? Well, one way is solar water splitting. Solar water splitting is what we call it when we use the energy from sunlight to split water into hydrogen and oxygen. In this way, we store the energy in the hydrogen bond for later use. I'll only focus on the hydrogen half of the water splitting, as oxygen isn't quite as interesting for us right now. First, we need to get our hands on some semiconductors, which have unique optical properties useful for this application. What I mean by this is that when a photon, a little energy packet from the sun, hits a semiconductor, it is absorbed by the semiconductor, and as a result, an electron in the semiconductor is excited to a higher energy state. Now, these two electrons have enough energy to help bond two hydrogen atoms together. We use two electrons to form one bond. Once the two hydrogen atoms are bound together in this way, that original energy from the photons is stored in this chemical bond. Once we have this hydrogen molecule, we can move it around, like transporting it on a truck or a ship, and we can store it. When we want to get the energy back out of that chemical bond, we can burn it just like any other fuel. For example, imagine a natural gas power plant. We can burn the hydrogen, and the power plant generates electricity, which is sent to our homes and businesses. But here is the super, really amazing, awesome thing. When we combust or burn the hydrogen with the oxygen in the air, which there's plenty of, we simply form water. This is much better than sending tons of carbon dioxide into the atmosphere where it hangs around and heats up the planet. Instead, solar hydrogen provides us with a way to satisfy our energy demands in a sustainable and environmentally friendly way, allowing us to fight back against climate change. That's all for a brief introduction to solar hydrogen, and now some jazz.
Hello there! My name is Centriole. That's a cool name, right? As if I'm the center of the universe. Wait, what? You never heard of me before? Well, that's pretty strange because I live inside of you. And I don't only exist in one place in your body, but I exist in a million copies. But hey, come back to me. Let's focus on me. I want to tell you about my life. Where do I live? Well, although my name sounds big, I'm actually very tiny and live in one of your cells. Among all the inhabitants in your cell, I was called the mysterious inhabitant because it is very hard to find me. Although I have been in your cells forever, I was only discovered about 120 years ago by Thomas Bovary, who must have had an eagle's eye to spot me inside my house, the Centriozone. I love my house. Don't you think it looks futuristic? It is round and has many layers. I live in the very center of the house, and depending on my needs, I rearrange my house to the way I want it. I'm actually pregnant at the moment. I have my child attached to me in a 90-degree angle. I know, it's a funny way of being pregnant. I'm a real family person, you know. In every cell of yours, I only have one close neighbor. She is not just my neighbor, but my family. It's actually my mother. So, do you want to know about my job? Well, it's a bit complicated, but let me explain. When you are growing up, your body makes more and more cells. Do you remember Mr. Nucleus? Mr. Nucleus's job is to store all the information a cell needs to live in the form of DNA. Almost like a library for all the knowledge you humans have. Now, when the cell decides to divide and multiply, Mr. Nucleus makes one copy of the DNA library. While Mr. Nucleus is really good at copying and sorting, he doesn't have the power to distribute the libraries into new cells afterwards. This is when my mother and I come into play. We are really strong, you know? We both rearrange our little house and take control of many strings, which scientists call microtubules. Then my mother and I use these strings to bring the DNA library from the center of the cell to our camps. But in the end, we both win. We make sure that every one of us has exactly one copy of DNA library each. At the end, we say goodbye to each other and go on to live our lives separately in different cells. But I don't live alone for a long time. I gave birth to my daughter, and she has now grown up to become a real woman. But I also have another job. Let's have a look at the origin of your life. I guess you already know about a sperm and an egg? Well, I hope so, because I don't want to have to tell you about the flowers and the bees again. Anyways, let's have a closer look at the sperm. I'm actually very important here. I make the tail of the sperm grow and help them move. And what about the egg? Well, guess what? I'm actually not there. During fertilization, I only come from the sperm. And this is because during the first cell division, it is important not to have too many neighbors fighting for your DNA libraries. But how did I disappear from the egg? Well, I wouldn't be the mysterious organelle if I told you everything.
Well, we hope you like the films, and now I would like to introduce Theo Blossom from the jury. He will share some reflection on the films and announce the winner. Theo?
Hi everyone, my name is Theo. I am one of the judges this year. Myself and the other judges really enjoyed watching all of the films. Lots of creativity, lots of very different way to create rich visuals using some very special techniques which take a lot of time and dedication, so hats off to that. The winner of the Swiss film is someone who created a film that is very pertinent to a current issue of energy creation in the world, and did a great job of bringing in some humour and some nice visuals to communicate the importance of help create energy. So the winner is Colin Firth with Solar Fuels. Well done, Colin. Bye, everyone.
Thank you, Theo. And now we should have with us Connor, the winner of the Swiss category of our SciFilmIt competition 2021. Hello, Connor. Congrats. So, welcome. I think we have some questions for you. Personally, I found your film very easy to follow and very, very entertaining. And I was actually impressed by how simply you digested such a complicated subject. And in particular, I found the voiceover extremely well done and super impressive. So do you have any previous experience doing the voiceover, or can you tell us more about the script writing process that you went through, something like that?
Yeah, well, first of all, thanks so much for the kind words, Eleonora. I appreciate it. In terms of the quality of the voiceover, I think what helped there was actually do a lot of music recording as a different hobby. And so I think the experience there really lent itself to having let's say, a focus on the audio quality and maybe a different perspective on that. In terms of the script writing, yeah, I took I've taken one excellent course at EPFL actually, and I think that really helps provide me a perspective about how to tell a story. You know, I know some of you and the audience members are familiar with this ABT philosophy, and that's what guided me in the script writing, I think. Did you have an excellent professor too in that course? There's a great professor, there was. Maybe that professor has a question for you too, Enrico.
I do have a question for you, Connor. Hi,
sure, Enrico.
Great seeing you. Yeah, like I wonder, like how you came up with the choice of having like a notepad as a canvas. Like usually people go for really blank white canvas, animation-wise. Mm-hmm. You stick with this really dirty looking, let's say, background.
Yeah, I think there was two reasons. One was more intentional, one was not intentional. One was, and what order they came in, I'll leave to mystery. But I think that for me, this grid paper, it's the paper we have everywhere around the lab. It's the paper we have in our offices. It's It's useful for graphing, for doing calculation. And so for me, using that paper was kind of like maybe an effort to bring the audience into this, kind of casual, more chalk talk kind of conversation about the topic, instead of having a more maybe formal, clean slate, as you say. But for that reason that we always have it in the lab and everything, that's also what I have around. And so I think it worked out, hopefully to bring the audience a bit more into the thick of it. Yeah.
Thanks.
So I'm not seeing any questions in the chat. So first of all, I'll say we have a couple of cameras from Nikon heading your way.
Wow,
congratulations, fantastic film. And I do have a question myself. So 1 hour of energy from the sun powers the whole planet for 1 year.
There's enough energy.
Okay. My question is, does this mean that, like, I don't know how much time, but does this mean that the inevitable future of energy is solar power?
I think, well, personally, it's solar. It's easy to say. It's objective to say that it's the most abundant energy source we have. So, what that means is, right, that when we harness it, we don't have to harness it very efficiently relative to maybe other energy sources, and we'll still have plenty of energy to supply our demands. But if you ask me, I think that the future of energy will be multifaceted, and maybe most importantly, it will be more decentralized from the way we have it produced now. And I think that would also result in a more equitable energy distribution, energy grid. So that's my political answer.
Thanks.
Okay, I think Enrico, you will follow the screening with our next session.
Yeah, congratulations again, Connor. And
thanks so much to all of you for organizing.
Sure, thank you. And so we now move on to our second and last section of the competition, which is the international section. So here we welcomed submissions from all over the world, and we're going to watch the 5 films that were selected. And the films are Don't Blame the Bets, Gravitational Lensing, How Tranexamic and Acid Stops Bleeding, Samsara, and Moonlight Slide. Enjoy.
Here we are, all together facing this global challenge. While the experts are trying to figure out an effective treatment or cure, the rest of the population listens. Although we're still learning about COVID-19, there's a widespread theory that points to bats as the potential origins of the virus. But are bats really to blame? Well, let's see what science can say about this. Scientists have described hundreds of different viruses in the coronavirus family so far, including 4 human coronaviruses which can cause the common cold. 3 of them, severe acute respiratory syndrome coronavirus, Middle Eastern respiratory syndrome coronavirus, and SARS coronavirus 2, are behind some of the new disease outbreaks that happened this century. Coronaviruses can be found in birds as well as in mammals, and very rarely some of these viruses can mutate and jump to humans, causing an infectious disease. In the current pandemic, the virus has rapidly spread among people. There are even cases where it has been transmitted from humans to pets and other animals in captivity. However, the original host of this virus is still a mystery. Like humans, bats are hosts to a number of different types of coronaviruses. However, none of those known to cause disease to humans, including SARS-CoV-2, have ever been detected in any of the world's 1,400 bat species. Some of them are similar, but have any of them had a role in the current epidemics? To know this, we need to track the evolutionary origin of the current virus. Scientists have compared the sequences of the complete genome of the new virus with other previously known coronaviruses. This showed that the current virus and others some bats carry are similar, but there are still many differences between them. The most similar bat virus found to date, and SARS-CoV-2, could have separated from a common ancestor around 40 years ago. Researchers have also analysed how the virus attaches to human cells. They have found that pangolins have a coronavirus with a membrane structure, similar to the current virus membrane. However, when scientists analysed its complete genome, the results showed that the pangolin virus is so distant to the present virus that pangolins couldn't have been the recent intermediate hosts. A scientific theory lays out that the current virus could have been present in humans for many years and recently mutated to become more capable of being transmitted. Did the current virus evolve from a bat virus? It's not as easy as it seems to know the evolutionary origin. The most likely link is that both viruses have a common ancestor, but is this really important? Understanding the epidemic origin is what matters more now. So, can bats transmit this virus to humans? At present, the evidence doesn't support this theory. Despite none of the presently known bat coronaviruses having ever directly infected a human, bats still have a poor reputation. Folklore of many cultures associate these mammals with a negative stigma. The fact that they are nocturnal and therefore mysterious means some people fear them, but the truth is that bats are extraordinary animals. When bats fly, their temperature raises to a fever-like level that could be harmful in other mammals. Bats use this to boost their immune system response and ward off the virus despite being infected. All the European bats feed mainly on insects, not blood. Some feed on spiders, and one species feeds on fruit. This brings many benefits to humans, like helping to control insects, some of which can damage crops and gardens, and some may even help control insects that can transmit diseases to humans, such as mosquitoes. COVID-19 has always been a human disease, and the host that infected the first human is still unknown. Regardless of the animal that transmitted the virus, culling bats is not the solution, and in fact has the potential of making a bad situation worse. If we can change the way we interact with nature, if we stop buying and consuming wildlife, if we preserve their habitats, we will be stopping future pandemics like this. Protecting wildlife is protecting us.
Our eyes crave baths of light, the flickering playground of shivering stars, an image of a blue arc on the rim, coiling around clusters of galaxies, the vivid shimmer behind you in the garden as the torch frames your silhouette in the dark. We long for a glimpse of planets in slow motion, counting them long into the balcony of the night. So after we see quasars in the distance, distorted, we want to understand how mass bends the light, how dark matter halos assemble over time by gravity, their complex webs cushioning around baryonic matter, or black holes infer their presence from distant stars or flowing accretion disks. Gravity flexes the structure of space-time, warping light from travelling in its straight line, as if a universal river pools at the sides of invisible stone. The brightness, lit from behind. When a large galaxy becomes the front view focal lens, far-off galaxies are magnified and curved, arching at the frames. Strong and weak lensing enhance surrounding or further set stellar hives. Some nurseries, billions of years in our past. If the foreground mass, background and observer are perfectly aligned, this Einstein ring resembles the imprint of a cereal bowl abandoned for morning play, a seemingly concentric stain. We try to see beyond what is immediately visible and illuminate what is known but concealed. Our bare eyes in the coldness of night, peering through a telescope, unable to locate most of the weight of the universe, missing out on all the things we cannot see.
When a blood vessel is ruptured and starts bleeding, the body responds by forming long strands of a tough yellow protein called fibrin that traps blood cells and plugs the leak. This is called clotting. However, at the same time, it triggers another process which is not always helpful. It begins with these playful canines called plasminogen, but they won't be playful for long. The torn blood vessel has released tissue plasminogen activator, or TPA as he likes to call himself, and he loves causing trouble. Once TPA shows up, the plasminogen dogs get nasty. These hounds of hell are plasmin. The plasmin dogs tear fibrin apart, making the bleeding much worse. Of course, plasminogen activator inhibitor 1, or PI1 for short, could put a stop to all this destruction, if she wakes up. And when PI1 does wake up, it's often too late. That's why we give tranexamic acid as soon as possible after bleeding starts. Tranexamic acid stops plasminogen binding to fibrin, sparing the clot from the plasmin dogs. and a nice blood clot can stop the bleeding.
Imagine you're an engineer and you're renovating the Centre Pompidou in Paris. All of this beautifully bent glass needs to be replaced and is about to go to waste. 195 tons of CO2 were emitted to produce this glass. Surely, you must know an architect who wants to reuse the glass. So, what's wrong with today's construction sector?
On n'aura plus accès de la même manière au même coût à certaines ressources et à certaines matières premières, et il faudra nécessairement qu'on trouve d'autres solutions pour construire. Le réemploi en est une, et ce jour-là, c'est mieux si le marché est prêt, quoi.
What is circular construction?
L'économie circulaire, c'est très simple, c'est quand les déchets des uns sont les ressources des autres et qu'on sort du schéma linéaire qui est ressources, utilisation et on jette. Et c'est qu'on vient prendre cette ligne et on la tord pour que le on jette redevienne la ressource des autres et qu'on soit dans un espèce de format comme ça circulaire et qui peut, s'il est suffisamment vertueux et suffisamment bien pensé, tourner à l'infini.
On est dans un système qui consiste à extraire, consommer, jeter. Et l'exemple classique, c'est la matière première. C'est-à-dire que quand vous construisez un bâtiment, vous fabriquez une porte en bois comme celle qu'on voit autour de nous, vous coupez un arbre, donc vous extrayez de la matière d'une forêt en l'occurrence, vous fabriquez une porte, elle joue son rôle de porte, puis un jour pour une raison X ou Y, on vient à démolir le bâtiment ou à changer la porte parce que c'est plus la bonne couleur ou elle correspond plus à telle et telle norme. Et puis à ce moment-là, on détruit la porte et on la jette et c'est la fin de l'histoire de la porte et de la matière qui la compose. L'économie circulaire, c'est l'inverse, c'est de dire on peut plus se permettre de jeter cette matière-là et on pourrait la réintroduire dans un cycle. Et finalement, cette porte, elle peut de nouveau servir de porte ou bien elle pourrait servir de table. Un petit bâtiment fait avec des portes de réemploi, qui sont des portes que vous voyez autour, sont les mêmes portes.
L'économie circulaire, moi je pense que c'est une opportunité formidable pour l'architecture, pour se réinventer, puisque c'est nécessaire de le faire aujourd'hui. L'opportunité aussi, c'est que ça va créer toute une série de métiers, puisqu'il y a des enjeux logistiques, de stockage de la matière, de son transport. On est rarement dans une congruité entre le moment où on va déconstruire et le moment où on va reconstruire avec cette matière, où on va la reposer dans sa nouvelle destination. Donc il y a aussi tous ces métiers-là, qui sont des métiers qui ne sont pas délocalisables.
Are you wondering what happened to the glass from the Centre Pompidou?
Le projet du Centre Pompidou, ça a été un alignement des astres qui, à notre niveau aujourd'hui, en 2020, etc., est assez rare. C'est-à-dire un projet, un besoin, c'est-à-dire un chef d'entreprise qui est venu nous voir parce qu'il voulait qu'on fasse des pavillons, des petites salles de réunion dans le hall de son siège social, et avec une correspondance en termes de timing, en termes de ressources, en termes de quantité, etc., un déchet disponible qui était les verres du Centre Pompidou. Et ça, l'alignement de ces deux facteurs, c'est ce qui a rendu possible, et c'est relativement rare aujourd'hui. Et c'est ce qui fait qu'on a pu aligner les deux et proposer pour les verres courbes de la chenille de Beaubourg une solution, une sortie qui évitait le destin tragique de la benne.
Young designers from Maximum reuse the glass with waste metal to make curved walls in a new project, saving tons of glass and steel this way. Considering the emblematic value of the Centre Pompidou, I'm happy we, collectively, went on a mission to find a new home for the glass. This is circular construction.
Eva Dadechan Moosehide Slide.
The Dawson City Landslide, also known as the Moosehide Slide, is a dominant feature of Dawson's cityscape.
Danoja people lived relatively peaceful lives in Trondequichin country until cannibals came along.
It is a prehistoric, pseudo-circular rock slope failure at the northern edge of town.
The people climbed the hill near where is now Moosehide to get away from the cannibals.
Dendrochronological analysis demonstrated an average movement rate of 4.5 centimeters per year.
They came upon the biggest tree in the forest, and this will be the one they used to roll down the hill to take out the cannibals.
This moving section of the debris could be characterized as a rock glacier or as an earth flow, although our present observations and measurements do not confirm either mechanism.
They thanked the tree for allowing it to be chopped down.
Earth scientists have long speculated about how the slide initiated and what the present mechanism of movement is.
Kapow! The tree starts a big landslide.
The Dawson City landslide involves two rock types: meta-sedimentary rock from the Yukon Tanana terrain and ultramafic rock on the Slide Mountain terrain.
The tree and the slide crashes down the hill towards the cannibal.
These units are interpreted as a volcanic arc assemblage overthrust by a sliver of oceanic crust.
The cannibals cannot escape. They are buried.
From the lobate shape of the debris deposit and the abundance of very coarse blocks, we believe the initial slope failure event was a catastrophic rock slide.
The people are joyful that the cannibals will not bother them anymore. They have big potlatch. They sing and dance.
The fractured nature of the outcrop has reduced rock mass strength and led to probable pseudo-rotational failure.
They also notice that the slide is shaped like a hide of a moose, and so the people called the place below the hill Moosehide.
Local inhabitants have wondered whether another catastrophic slide will ever occur and whether the debris will continue to encroach on their property.
All right, so we watched 5 great films. Congratulations to all the participants. The jury had a really tough time deciding the winner due to the high quality, as we just saw, of both the storytelling and the fascinating science that has been communicated. The film that they chose as the 2021 SciFilmIt competition Best Global Film was a film that impressed them in all areas. The story was simple and clever, and the visuals were beautiful. So congratulations to Hywel Roberts and his film How Tranexamic Acid Stops Bleeding. And we should have Hywel here for a quick chat. Hi, Hywel. I can see he's connected, but I cannot neither hear him or see him. Hi, Hywel.
Hello.
Here you are. Thank you, Hywel. Thank you for joining us. Congratulations.
Pleasure. Thank you.
See he's connected, but I cannot. And we have a couple of questions for you, like if you don't mind.
Oh, hang on one moment. I just need to. I've got the YouTube on at the same time. Hello.
Here you are. Thank you. All right. Okay. Now seems great.
Apologies.
Okay. No worries. Thank you. I have a question about the film that you made, and which is like, I was really impressed by the balance of the artistic and the scientific components of the film. And I wondered then how the collaboration happened. Like, how much did you interact with the, I think it was the School of Hygiene in London, if I remember?
Yes.
Like, was there a long back and forth? Were you just given an input at the beginning and you went on your own road?
There was quite a bit of back and forth. I was fortunate that the person who approached me to make the film is an uncle of mine, Professor Ian Roberts, who works at the school, and we collaborated with each other before on a shorter animation, again to do with tranexamic acid. Except this one we were going in a lot more depth into the process, and so. Yeah, first he approached me, we sort of talked about it, and then I was given a brief, and I sort of did some research into it, and came up with some ideas, and then we met and discussed those ideas, and really, like for example, the idea of the dog and the ball that came from when I was at my uncle's house, and we were watching television with his young kids. And then there was a dog and a ball on the television, and that idea came up. So it was very close kind of collaboration in that way. And then throughout the script and creating the storyboard animatic, we were constantly, on the phone and sending work and back and forth to each other. And it was one of the trickiest things was as you say, finding the balance, because there'd be things that would work for the story but wouldn't work in terms of the science making sense, and vice versa. And so we'd get into these really head scratching moments, like, okay, how do we solve this? And that was really fun, and I'm happy to hear that you think it has that balance, because it was a struggle to get that.
Yeah, absolutely. I wonder if Robbie and Elio have.
Yeah. So congratulations, of course. It's a fantastic film. I was going to ask you about the analogy, but you've kind of answered my question. I love it. It fits really well. You have this cute character. But I think another thing that I was really impressed by is that you created a world that I was immersed in. I was in that, under that skin. I was with the bleeding blood vessels, and this is so tricky in such a short amount of time, and we so often try to pack so much in, and then we lose something there. And I think you really found a balance, as Enrico was saying, that meant that we were immersed in a world that felt real, that was cute, that worked really well. So first of all, I want to congratulate you on that. Particularly loved the Pi 1 character, who really was another, like, kind of, adding another character can be really risky, but it just felt right. And so, congratulations. I don't know if you want to add anything to that.
Oh, thanks very much. And yeah, I'm glad to hear you felt that that it worked visually and yeah, the Pi1 character went through many different kind of different iterations, and then we finally found something that worked. And like as you say, it's right. It does. The film wouldn't be the same thing without it. I mean, it'd be very difficult to explain the process without that character and as well. Yeah, as for the inspiration for the visual style, I really wanted to keep things simple because the information was so complicated and it had to be really clear and concise. And so I looked at one of my favorite artists, picture book artists for children, is John Classen, and he has a very simple style. And other picture book illustrators as well, and animations. So I was inspired by that really, and just creating this world and just keeping it, the only thing that you see is what's needed to tell the story and explain, how tranexamic acid works.
Fantastic, thanks.
So my question will be, as 2021 SciFilmIt competition winner at this point, what would be your suggestion or advice for future scientists that want to approach the script writing for short films? What would be your secret, if you can share it, or your advice?
So yeah, I guess my advice for anyone coming from a scientific background would be, I guess, to learn about story as much as they can, and if, for example, they want to make the film in animation, to learn about the process of animation as well. But in terms, as you're talking about script writing, the story is very important, and having that doesn't have to be an expert understanding, but kind of a basic understanding of stories and how they work is really helpful, both in kind of communicating the idea, if you're working with an artist or if you're working on your own, coming up with a script. Yeah, because I did a script writing course a few weeks before I started the project, and that was really helpful just to find a way of getting the science to work in a story in a way. It's kind of, because it's not always straightforward. So my advice would be to learn about story and to really, as a scientist, to really know and understand the science, which they would, and because those two things together is and that's why I think the collaboration between myself and my uncle has always worked quite well, is that he's he knows the science really, really well, and he's also a very good communicator. He's a great storyteller, and then I come from the animation and storytelling background as well, and that kind of fusion helps make it work.
Thank you. Congratulations.
Thank you.
And I guess this brings us to the end.
Oh, we have a question actually from the YouTube.
Oh,
so it's actually from Connor. So I mean, I'm guessing, Hywel, that you're a trained animator, or am I? Yeah. So Connor's question was, what software did you use, and but how did you do the animation? Which is quite a broad question, but I guess you started by sketching, but you know better than me.
Yeah, no, I started sketching in my sketchbook after reading the information. And a lot of writing as well. I worked just a lot of little notes, mix of both. And then for the animation, after we created the whole film, and you can see it online actually, is done first in a storyboard. So that's drawings which kind of illustrate the script in a way, visualize the script, and that's timed out, with sound and everything, and that is essentially the film. I see the animation after that's just the coloring in really, and then for the animation software wise, I used a piece of software called Moho Pro, I think it's called, and it's a great bit of software. It's not too expensive, and And yeah, you can create this kind of cutout style and a mix of other styles in it, and it worked well for this. Yeah.
Thank you, Hywel, for answering all the questions, and congratulations again on your.
Thank you all for organizing this and selecting the film. It was really great to be part of.
Thank you.
Congratulations. Bye-bye.
Bye.
Oh, wow. So those are the 8 films and the 2 winners. It was really wonderful to have people asking questions and to meet some of the creators and to see them all again, actually, because they're all fantastic films. So that's where we are. It brings us to the end of the first ever SciFilmIt competition. It's been amazing to receive all the films, as well as the ones that were selected, and to celebrate them all tonight. So SciFilmIt kind of goes on, and we have kind of quite a few things coming up in the next few weeks. Next week, we actually have a live show in Lausanne. So we have a live cinema show where we'll show 6 films made in Lausanne. And yeah, they're fantastic. I've already seen them. There will also be a link to watch them online. So if you're not in Lausanne or if you're not nearby, you can watch them online. We then will have a hackathon in Geneva the week after. And then once more, we'll have a cinema event for that hackathon. So we should see 6 more films created in that hackathon. Once more, thank you so much to the amazing jury, and thank you so much to the University of Geneva and Nikon for their support. It's incredibly important. Thank you to Enrico, thank you to Eleonora for being wonderful co-hosts with me, and thank you to the audience for being for watching this. It really it means a lot to us, and if you're looking to get involved, get in touch. Lots of different ways to get in touch. Email and social media, great ways. And so thank you, and have a really nice evening. Goodbye.